Hello, my name is Mike Horton. I am a second-year genetics and genomics major. I work in the COP lab, and over the summer of 2019, I worked under the guidance of Dr. Emily Delaney in determining whether there is a fitness effect of color polymorphism in Drosophila serrata via a heat knockdown assay. Throughout this video, please comment and ask questions. Almost all species have polymorphisms present in their populations. For example, these snails all look different but are considered to be the same species. These peafowl show an example of sexual dimorphism, where one sex, typically the males, look much different from the other. Sexual dimorphism is used as a way to attract mates and for females to assess fitness of potential partners. Polymorphisms typically appear in populations as spontaneous mutations. These are selected for over time if considered advantageous. The maintenance of polymorphisms in animals is not well known. However, one of the first things to consider when investigating maintenance of polymorphisms is determining why they exist and how they affect an organism's fitness. For this example, I used Drosophila serrata as a model organism. Polymorphisms is seen in this species as a difference between sexes, but Drosophila serrata are a bit different than most sexual polymorphs. In the left photograph, we see homozygous light flies. Females on the left and males on the right. Both appear light, which makes sense. In the right photograph, we see homozygous dark flies. Again, the female is on the left and the male is on the right. Only the female has the dark phenotype, while the male is light despite its dark genotype. In Drosophila serrata, all males have a light phenotype regardless of their phenotype. This is called a female-limited dimorphism. So, what is FLCD? Well, FLCD is found in about 20 members of the Drosophila montium subgroup. Drosophila serrata, Kikowai, Leontia, and Burlei are distantly related in this subgroup, but FLCD was mapped to the same locus in the transcription factor POU domain motif 3, otherwise known as the PDM3 locus, which acts as a repressor of abdominal pigmentation. Serrata typically have a structural variant in the first intron as displayed in this figure. This information is from the Yasin and Delaney 2016 paper. So, knowing that Drosophila serrata have FLCD, we wanted to know why these two female morphs are preserved in the population. There is a gradient through Australia of dark and light females. Darker females are typically found at a higher frequency in the southern regions of Australia. These regions are cooler and more moist, while light females are found at a higher frequency in northern regions, which are typically hotter and more dry. So, do these phenotypes or genotypes have an effect on the fitness of flies in these different climates? Well, first off, we predicted that because light females are found at higher frequencies in the northern hotter climate, homozygous light lines will have a higher fitness in hotter environments than homozygous dark individuals. This was thought because lighter females will affect more light and heat, thus having a higher heat tolerance than dark females. Since males do not show this dimorphism, all males were predicted to behave the same as light females. This fitness was measured via a heat knockdown assay by comparing heat knockdown times between different light and dark lines. For each line, we had 9 dark and 9 light. 2 females and 2 males were selected from main stocks under CO2 gas and placed in a vial with food for 3 days and left to breed. After 3 days, adults were removed and offspring were left to mature and enclose. This was done to control population size of larvae within each vial. Virgins were collected under CO2 gas every day and left to age for 4 days after collection. Once 4 days had passed, individual flies were placed in glass tubes with sealing caps using an aspirator in sets of 8 and positioned in a plastic and foam rack that had equilibrated in the 39 degrees Celsius water bath, as seen in this picture. A timer was started and a heat knockdown time, which was when flies fell over and remained upside down for at least 10 seconds, was recorded. Knockdown times were recorded for 20 males and 20 females for each strain. Here is all 18 lines represented as a mean of individual knockdown times for each sex. As you can see, there's a general trend of dark lines having higher means than light lines. Next, each color line was combined for each sex. Here it is a bit easier to see the separation of knockdown times for each color. Looking at just these box plots, the medians of dark males and females follow both upper quartiles of light males and females. Interestingly enough, despite this dimorphism being limited to females, dark males have similar knockdown times to females, despite not phenotypically being dark. However, just looking at a box plot is not enough information to come to a solid conclusion. This data was then run through ANOVA using a linear model due to the nesting of individual knockdown times within sex, within lines, within color morph. The three interactions were males versus females, light versus dark, and sex and color combined as an interaction. Here you can see a p-value of 0.09899 was obtained for color. This was the lowest p-value found when comparing all interactions in this model. This interaction, or the difference of knockdown times between light lines and dark lines, was the largest, but a p-value of 0.05 or lower is typically considered statistically significant. However, after discussing with my mentor, Dr. Emily Delaney, my PI, Dr. Kopp, and Dr. McIntyre from the University of Florida, we decided to use a survival curve analysis to better understand the data. Here is a survival curve showing the difference in dark and light genotypes. After performing a log-rank test to test the difference between light and dark lanes, we got a p-value of 4 to the negative 14th power. This rejects the null hypothesis of dark and light lines having the same survival probability in this heat assay. Now that we have sorted through this data, how does it relate back to our hypothesis? First off, there was actually a contradiction from what was first hypothesized. Dark lines, on average, had higher heat knockdown times for both sexes compared to light lines. We can interpret this as heat not being the only factor affecting fitness in this dimorphism. Future experiments would be to test desiccation resistance, chill coma recovery, and UV light resistance. If you have any other variation in climate that can be tested under lab conditions, please comment in this video. Over the summer, if undergraduate research is permitted, I plan to test desiccation resistance in a similar manner, but with a higher replication. Also, what we interpret from this data is that genotype matters over phenotype. Homozygous dark males performed at the same level as homozygous dark females despite having a light phenotype. This leads to the idea that the allele controlling pigmentation at the PDM3 locus may be causing another difference besides abdominal pigmentation. The light allele is 1 kb in length compared to the dark allele, which is 1.5 kb. This difference in allele length may affect nearby regulation sequences for other genes or something else. Since light is dominant over dark, it would also be interesting to look at heterozygous flies and see if they have knockdown times like light lines or an intermediate knockdown time. Since the dimorphism is restricted to females, but the fitness effect of heat tolerance is not, some other factor may be involved with the maintenance of this female's limited dimorphism, as well as the fitness differences between these two genotypes, regardless of the fly's phenotype. Thank you so much for listening to my presentation, especially in this novel format. I look forward to pursuing the question of possible other fitness differences, as well as why males do not display this dimorphism, but appear to have similar fitness as their female counterparts. If you have any comments or questions, I would love to read them. A shout out to my amazing lab and everything I've learned from them. Please enjoy the rest of this online conference, and thank you for listening.